Dear viewers, greetings. In this video, we are going to see about the S-shaped curved rod Campylobacter jejuni. First, general characteristics of Campylobacter jejuni. The Campylobacter jejuni is a gram-negative, thin, comma-shaped or S-shaped curved rod, and it is motile in nature due to the presence of bipolar amphitrachous flagella and its size ranging from 0.2 to 0.9 micrometer wide and 0.5 to 5 micrometer long. And this Campylobacter jejuni belong to the family Campylobacteriaceae. Capsules absent and endospores also absent. The mode of respiration observed in the Campylobacter jejuni is microaerophilic respiration, and it requires very low amount of oxygen for its growth. The optimum temperature required for the cultivation of Campylobacter jejuni is 37 degrees Celsius to 42 degrees Celsius, and optimum pH is 6.5 to 7.5. And regarding habitat, Campylobacter species are widely distributed in most warm-blooded animals. They are prevalent in food animals such as poultry, cattle, pigs, sheep and ostriches, and in pets including cats and dogs. The bacteria have also been found in shellfish. A total of 32 species and 13 subspecies are now recognized in Campylobacter species, many of which are associated with human diseases, but only 4 species like Campylobacter jejuni, Campylobacter coli, Campylobacter fetus and Campylobacter absaliensis are common human pathogens. The bacterium Campylobacter jejuni was discovered by the German pediatrician Theodor Estrich in the year 1886 from diarrheal samples of children. Disease transmission of Campylobacter jejuni. The main route of transmission of Campylobacter jejuni is generally believed to be foodborne via undercooked meat and meat products as well as raw or contaminated milk. Contaminated water or ice is also a source of Campylobacter jejuni infection. Campylobacteriosis is a zoonosis, a disease transmitted to humans from animals or animal products. Most often, meats are contaminated by Campylobacter jejuni during slaughtering. Finally, in animals, Campylobacter species seldom causes disease. Virulence factors of Campylobacter jejuni. The ability of the microorganisms to cause an infection is called as virulence, and the factors responsible for causing an infection in human beings is called as virulence factors. And here Campylobacter jejuni have 3 virulence factors. The first virulence factor is adhesins, the second virulence factor is cytotoxins, and the third virulence factor is enterotoxins. An incubation period for Campylobacter jejuni infection is 2 to 5 days, and the infective dose required to cause an infection is 800 organisms. Pathogenesis of Campylobacter jejuni. Infection with Campylobacter jejuni results from the ingestion of contaminated food or water, and the infective dose can be as low as 800 organisms. To initiate infection, the Campylobacter jejuni must penetrate the gastrointestinal mucus, which it does by using its high motility and spiral shape. The bacteria must then adhere to the gut endocytes, and once adhered can then induce diarrhea by toxin release. Campylobacter jejuni releases several different toxins which vary from strain to strain, mainly enterotoxin and cytotoxins, and this correlate with the severity of the enteritis. During infection, levels of all immunoglobulin classes rise. Of this, IgA is the most important as it can cross the gut wall. IgA immobilizes organisms, causing them to aggregate and activate complement, and also gives short-term immunity against the infecting strains of organisms. The other immunoglobulin classes act on the bacteria entering the bloodstream, that preventing bacteremia. And Campylobacter jejuni can also stimulate the cellular immune system. By this seems to play only a small role in preventing infection. And finally, Campylobacter jejuni antigens that cross-react with one or more neural structures may be responsible for triggering the Guillain-Barré syndrome. The Campylobacter jejuni is responsible for causing two clinical diseases. The first clinical disease is gastrointestinal infections and the second clinical disease is Guillain-Barré syndrome. The first clinical disease caused by the Campylobacter jejuni is gastrointestinal infections. Gastrointestinal infections with Campylobacter jejuni is most commonly as acute enteritis with diarrhea, fever and severe abdominal pain. Affected patients can have bloody stools. The disease is generally self-limited, although symptoms may last for a week or longer. The range of clinical manifestations includes colitis, abdominal pain, appendicitis and bacteremia. Finally, chronic enteric infections may develop in immunocompromised patients or patients with AIDS and be difficult to treat. The second clinical disease caused by the Campylobacter jejuni is Guillain-Barré syndrome. Guillain-Barré syndrome is a rare but serious autoimmune disorder in which the immune system attacks healthy nerve cells in peripheral nervous system. Guillain-Barré syndrome often begins with tingling and weakness starting in the feet and legs and spreading to upper body and arms. And finally, Guillain-Barré syndrome progresses. Muscle weakness can evoke into paralysis. Laboratory diagnosis of Campylobacter jejuni. The first diagnostic method is microscopic examination. Under gram staining, the Campylobacter jejuni was observed as pink, thin, comma-shaped or S-shaped curve gram-negative rods. In motility test, motile cells were observed due to the presence of bipolar amphitrachous flagella. Next, colony morphology on culture medium. Blood agar plates containing antibiotic is the media of choice for Campylobacter jejuni cultivation. Inoculated plates are incubated at 42 degrees Celsius in a microaerophilic atmosphere containing 5% oxygen and 10% carbon dioxide. In mechanical agar, Campylobacter jejuni was observed as pink colored lactose fermenting colonies. Next, biochemical test. Catalase test positive, oxidase test positive, urease test negative, indole test negative, MR test or methyl red test negative, VP test or Voges-Fluoroscore test negative, citrate utilization test negative, nitrate reduction test positive, and finally hydroxide hydrolysis test positive. 16S rRNA sequencing and polymerase chain reaction are the two molecular diagnostic methods used for the diagnosis of Campylobacter jejuni. Finally, treatment and prevention. For gastroenteritis, infection is self-limited and is managed by fluid and electrolyte replacement. Severe gastroenteritis and septicemia are treated with erythromycin or azithromycin antibiotics. And finally, gastroenteritis is prevented by proper preparation of food, consumption of pasteurized milk, and preventing contamination of water supplies. Dear viewers, that's all about the bacterium Campylobacter jejuni. Thank you for your support. Thank you.